Hello, dear students. So we are again back to our class protein synthesis. So I hope you have gone through my yesterday's video. Let us move now to continue the topic protein synthesis. So you can see this is the pathway of making a protein, that is DNA is converted into mRNA, which is termed as transcription, and mRNA along with tRNA and ribosomes gives us the protein, which is called translation. So transcription and translation are the two major steps of protein synthesis. The production or synthesis of polypeptide chains is the protein. There are two phases, transcription and translation. mRNA is produced in the nucleus of the eukaryotic cell, and this mRNA is produced in the nucleus from DNA, and this process is called transcription. And mRNA, after transcription, mRNA goes to the cytoplasm and produces protein. So this is the central dogma of protein synthesis. So this is a eukaryotic cell, diagrammatic representation of a eukaryotic cell. Here you can see in the nucleus the DNA, which is having the information in the form of nitrogenous bases. This DNA is transcribed into pre-mRNA. So this pre-mRNA is a unprocessed RNA. It is not yet functional. So after transcription, the pre-mRNA is processed to form mRNA, and the mRNA is the functional mRNA. Now the functional messenger RNA will leave the nucleus through the nuclear pore, and it will come to the cytoplasm of the cell, where it will attach with the ribosomes present in the cytoplasm. And you can remember, ribosome is the site of protein synthesis, and in the ribosome in attached form, with the help of tRNA, it will connect the amino acids as per the genetic code present in the mRNA, and a long chain of amino acids will be formed with the help of peptide bonds, and this chain is called polypeptide chain. So polypeptide chain is the protein. So in this way the protein is synthesized. Now let us see what is transcription. We have seen transcription very simply, so try to understand this. Transcription means it is the conversion of DNA into mRNA, or mRNA synthesis. So as we know that DNA is a double helix. It has a 5' or 5'3'3' strand, and 3', 5' strand, two strands. One is top and the other is bottom. So the top strand is the coding strand, and bottom strand is the antisense strand or template strand. So during the transcription, the DNA double helix, the double stranded structure of DNA is broken. That means the double stranded DNA is unzipped, and the 5', 3' strand of the DNA, it becomes the mRNA. Only the difference is, when it becomes the mRNA, the nitrogenous bases T is replaced by U. So you can see in the DNA the 5', 3' strand is starting with the nitrogenous base ATGCC, and the mRNA, which is produced, which is synthesized from the DNA, having the 5', 3' end with the nitrogenous bases AUGCC, like that. So now after the transcription from the DNA segment shown in the screen, the mRNA is produced or synthesized. Now transcription is a process where the DNA double helix or the double stranded structure of DNA is broken or unzipped, and it is a enzymatic process where RNA polymerase is utilized for unzipping the DNA strands. So during transcription, RNA polymerase binds to DNA and separates the DNA strands. RNA polymerase then uses one strand of DNA. That means you have seen in the earlier slide that the top strand is used to make the mRNA. Now promoters are regions on DNA that show where RNA polymerase must bind to begin the transcription of RNA, and that is called Tata box. And specific base sequences act as signals to stop, and that is called termination signal. So RNA polymerase will help in start of transcription, and the promoter are the sites or regions where the mRNA transcription will be starting, and there are some specific base sequences which are called terminal signal where the transcription will be stopped. So let us see with this diagram. So this is the orange color, this is the RNA polymerase enzyme, and the DNA where the RNA polymerase attaches, the double helical structure is broken, and the top strand is over the RNA polymerase, and below is the bottom strand which has complementary nucleotide in the mRNA, and it is pairing with the mRNA nitrogenous bases. So the mRNA is produced in right direction of transcription. Now after the transcription, that is mRNA, when mRNA is produced, that mRNA processing is required before going for translation. So after the DNA is transcribed into RNA, editing must be done to the nucleotide chain to make the RNA functional. So here we should know that there are some structures, or there are some segments present in the DNA which are called introns. So those introns are actually non-functional segments of the DNA, and that non-functional segments are required to be sniped out, or it is required to be eliminated from the mRNA, because it will be having no function in codifying an amino acid. So the introns are sliced out or sniped out. Again, we know that exons are the segments of DNA where the code for proteins are there, and these are actually coding the different amino acids for synthesis of protein. So exons will be, again, they should be put together in the mRNA. A guanine triphosphate cap is added to the 5' end of the newly copied mRNA, and a poly A tail is added to the 3' end of the RNA. So when the mRNA will be having only the exons, that means the coding segment of the DNA, then at the 5' end there will be a guanine triphosphate cap, and at the right side at the 3' end there will be a poly A tail at the 3' end of the RNA. Now the newly processed mRNA can then leave the nucleus. So after this packing of exons, by guanine triphosphate cap at the 5' end and the poly A tail